Hello, my name is Jordan Balson, and I'm a grad student in the Lefebvre Wong Lab at Queen's University, and I'm here with the River Symposium to talk to you about my research on assessing the potential of Caenorhabditis elegans and the bioremediation of Microcystis aeruginosa. So to get started, as a bit of a background, cyanobacterial harmful algal blooms, or CHABs, are increasing in prominence and severity across the world due to climate change and factors such as increased temperature, elevated CO2 levels, and nutrient leaching from fertilizers that causes eutrophication. So cyanobacteria, more commonly known as blue-green algae, are photosynthetic prokaryotes that can produce dangerous secondary metabolites known as cyanotoxins. And these toxins can impact human and animal health and overall have devastating impacts on freshwater ecosystems. CHAB damage also has a monetary cost and can affect things such as tourism and recreational opportunities. It can have a bioremediation cost, and it has an overall loss of existence value. So when we think about bioremediating these CHABs, we think about the abandoned mystery snail, Viviparus georgianus. So Viviparus georgianus is a suspension feeder. So what that means it does is it ingests cyanobacterial cells, which it will filter feed, and it will then either re-ingest them or disregard them. So when it's filter feeding, it either ingests and digests the cyanobacteria cells, which it comes out as feces, or it can ingest them, disregard them into their pseudofeces stack, and drop them. Either way, whether it's feces or pseudofeces, partially digested and partially undigested live cyanobacterial cells are translocated to the benthos. However, once they're on the benthos, this isn't a complete solution. So it partially fixes the problem because it keeps them from reaching their disruptive colonial blooming phase. However, eventually these live cells that are still in the pseudofeces and feces are able to be liberated from their feces or pseudofeces due to currents. And from there, they're able to form and rejoin blooms and go back to their disruptive colonial phase, where they produce the most toxins and are able to divide at the most explosive rate. So overall, we need another candidate that will take care of the bioremediation in the benthos. So nematodes, such as Caenorhabditis elegans, are excellent potential candidates for this bioremediation. C. elegans is a bacterivore that has several detoxification pathways, such as the phase 2 detoxification system. So Microcystis aeruginosa, a species of cyanobacteria, produces a cyanotoxin called microcystins, which is a hepatotoxin and a mild neurotoxin. So this phase 2 detoxification system actually has the potential to target microsystems, and this pathway has the potential to provide tolerance and possible detoxification to these toxins. So overall, I hypothesize that C. elegans and other aquatic nematodes are viable organisms for bioremediating CHABs in combination with Viviparus georgianus. And I hypothesize that nematodes will be able to ingest, digest, and detoxify microcystis cells contained within snails' pseudofeces and feces. So overall, I'm going to be testing this by analyzing C. elegans' ability to ingest, digest, and detoxify microcystins. I'm going to assess its impact on overall C. elegans' health, stress response, longevity, and their feeding behavior. I'm going to determine C. elegans' ability to detoxify microcystins and overall determine its ability to ingest pseudofeces and feces that contain toxic microcystis cells. So looking at my preliminary results, we first see fat accumulation. Fat accumulation was done with an oil red O fat stain, which stains neutral triglycerides, and we were able to see what fat they accumulate based off of their diet from that. So as you can see, toxic microcystis and OP50 as a diet both were able to accumulate fat and metabolize their food and incorporate it into their bodies. And there doesn't seem to be a significant difference in the amount of fat that they accumulated. Looking on further at longevity, the only diet that seems to impact overall longevity is whole toxic microcystis, and this could be due to mucilage production, which isn't completely understood, but is thought to have a role in protection against predation. Looking at figure 5 is their Dower stress response. And as you can see, diet did not appear to impact their Dower stress response. Looking at pharyngeal pumping and their thrashing response, neither was significantly different depending on their diet. So overall from that, we're able to interpret that there was not a significant impact from diet on their overall health in terms of their pharyngeal pumping, which is their feeding behavior, and their body thrashing, which is their overall mobility and health. Looking at their ability to detoxify microcystins, we see that in the presence of worms, there is an immediate spike in microcystin, which makes sense since microcystins are released upon cell death, and then it slowly decreases. In the presence of mutant worms, there's the wild type worm and there's the mutant QV224 worm, which is an SCAN1 knockout, which is an essential transcription factor involved in their stress response pathway or their phase 2 detoxification pathway. So looking at the first graph, you can see that there's immediate increase in microcystins in the presence of worms, which makes sense as it's hypothesized that microcystins are released as cells lyse, which they would be as they are ingested by the worms. So you see an immediate spike in microcystin concentration, and then it goes down. In the second graph, again, you see the immediate spike in microcystin concentration, and then it slowly goes down. And in that, there's the two worms, there's the wild type worms and the mutant SCAN1 knockout worms. And as you can see, the wild type worms do seem to have their toxin amount decrease more quickly than the mutant SCAN1 C. elegans. So this suggests that the detoxification, or its ability to decrease the microcystin concentration, might be due to the SCAN1 protein that's involved in the phase 2 detoxification pathway that the SCAN1 knockouts do not have. So overall, looking at my data, longevity might have been impacted by a diet of whole toxic Microcystis, but a diet of toxic Microcystis did not appear to impact their dour stress response, their locomotion, or their feeding behavior. For fat accumulation, they were able to ingest, digest, and metabolize Microcystis. And for the ELISA, C. elegans could possibly detoxify microcystins, and the SCAN1 protein could be involved, but overall, the natural degradation and growth rates of microcystis make this reason difficult to deduce. And for pseudofeces and feces, they likely have the potential to ingest and digest it, but we need to do further experimentation to confirm that. Thank you very much for listening.